So last week we spoke about, not last week, on Monday, we spoke about the primers and the enzyme called as a primase, right? A quick review. Uh, we said that primase is an enzyme, a very important enzyme, okay, which is required for the synthesis of a short primer. Okay, what is a primer? Primer is like a small oligonucleotide, like maybe 4 to 15 nucleotides long, sometimes even longer than that, like 100 to 200 nucleotides, right? And then we said this primer is very much required because your DNA polymerase will not be able to add the nucleotides unless and until there is a primer available. That's what we learned, in the previous class, right? So it needs something to latch on, okay, and that is basically the primer, okay. And primer is basically giving the free dash, free 3' hydroxyl ends, like, so it's providing these ends, and nucleotides are being added to these 3' OH ends constantly, and the chain is being extended, as you can see over here. Okay, so the green one is a primer, and then this is a leading strand, and this is a lagging strand over here. Okay, and one more important thing which you need to remember is that primer is never made up of, DNA. It's always made up of ribonucleotides. It's made up of the RNA. Okay. So moving on further, so if the question says that what is an RNA, why is RNA primer necessary for DNA replication? We know that DNA polymerase can add nucleotides only to an existing strand. Okay, and that existing strand provides us what? 3' OH ends. Okay, so that the nucleotides can be added to it and it will be extended. Okay, is it clear about the primer till now? Are we okay? Okay, now let's talk about a very, very important enzyme called the DNA polymerase. Replication would never ever exist without DNA polymerase. Like, DNA polymerase is the important enzyme. Okay, so it is the enzyme which can synthesize the new segment of the DNA. Okay, and if you remember, I told you that it only works in in 5 to 3' direction, right? We spoke about that, the directionality, read up and write down, right? So it can read the DNA strand only in 3' 5' direction, okay, but it can write down only in 5 to 3 direction. Okay, that's what we learned previously, right? So DNA polymerase is like the most important enzyme because it's the only enzyme which can form this new complementary strand of the DNA. Okay, and we just spoke about it. It needs a primer. That is like a very important thing to remember here, right? It needs a primer. Without primer, it cannot extend the DNA strand. Okay, so, and it always try to put the nucleotides in such a way so that adenine, if in a parent strand we have adenine over here, so what will be there in the new strand, guys? We will have, in the new strand, the DNA polymerase will try to incorporate which nucleotide, which
Thymine.
nucleotide? Thymine. Okay, wonderful. If we have guanine over here in the parent one, here it will be the cytosine, right? That is very, very important. So whatever is present on the template, right, the complementary base will be put by the DNA polymerase on the other strand. So base pairing rule will always be observed. Okay, remember that. Okay. And so this DNA polymerase, basically, once it incorporates the base, and then you know that we have a phosphodiester bond in the sugar phosphate backbone, right? So this DNA polymerase is very important because it can form the the phosphodiester bond between the nucleotides. That's how the nucleotides are being added. Okay, that's how it joins the sugar and the phosphate backbone, right? And we already spoke about, in the lagging strand, they are produced in short fragments called as Okazaki fragments. We already spoke about that, right, previously, the lagging strand. Then the last step would be the ligation and the DNA ligase. What is a DNA ligase enzyme? This DNA ligase enzyme is like, glue stick. The way your glue works, right? If you have to stick, something, if you have to stick your two, let's say whatever it is, right? Two sticks, you will put a glue over here, you put a glue over here, and these two sticks will be They will stuck to each other. So what will happen basically? So ligase, DNA ligase is like your glue. Remember that it's called as a molecular glue, right? Why molecular glue? Because it helps in joining the DNA fragments or RNA fragments, whatever fragments we have, right? So where do we need to join? We first need to join the Okazaki fragments, right? We know that Okazaki fragments are produced in, in smaller fragments, okay? So what is to be done first? First of all, the primase, which is the primer, which is synthesized. Let's say this is a primer here. These are Okazaki fragments, but every fragment has a primer, right? So every fragment will have a primer, okay? First of all, the primer has to be knocked out. We need to remove the primer, right? So primer is also removed by the DNA polymerase, we're not going to details of that, because if you go into details, then it's different. It works differently. The exonuclease activity works differently in prokaryotes and eukaryotes. We're not going to details of that. We'll just have a generalized concept, because the concept is totally different in eukaryotes and the prokaryotes. Okay, let's say that we have we call it an exonuclease activity. Okay, so what is exonuclease means? Removing something, knocking out something. Okay, so this will be knocked out because we don't need these primers now, right? The fragments have been synthesized, so we need something to which can erase it. Okay, so the way I'm using, oh sorry, I erased everything out. Okay, let's say these are Okazaki fragments, right? And we have here small green ones are the primers, okay, which were initially formed, right? We just need to remove the primers here, right? At the end, once the DNA strand is synthesized. So how will you remove the primers? Okay, for example, I'm using an eraser here, right, to erase this. So similarly, DNA polymerase will have an exonuclease activity, like a cutting activity, trimming activity. So it's going to trim these primers, okay? But as the primers are trimmed, don't you think, say, there will be a gap created, isn't it? There will be a gap. So look at this. There's a gap between this Okazaki fragment and this Okazaki fragment, okay? There'll be definitely a gap created over here, right? So what has happened, the space which was filled with the primer initially, right? So then the DNA polymerase will try to put, the fragments over here, right? So that this is also filled, but we still have a gap to join the two Okazaki fragments, right? So the two Okazaki fragments will be joined by the, by the DNA ligase. They'll be first filled with the, the nucleotides, and then the joining reaction will be taken care by the DNA ligase. Okay. So again, a very, very important enzyme, okay, because joining the nick means a gap, nick means a cut, right? So you should be very much, orient yourself towards these molecular, vocabularies which we use in molecular biology or in biochemistry. So if you try to, if you see a word like nick in your paper, so nick means gap, okay, so NICK. Okay, so DNA ligase is the enzyme which joins the Okazaki fragments and joins the nicks, right? The gaps present. Okay. Now that was all about different types of enzymes. Okay, and the only enzyme which I think did not discuss was a single-stranded binding proteins. I don't think so. That's mentioned over here, but I think that's mentioned in the table. Did I speak about single-stranded binding proteins? Anyways, we have something called as a single strand binding proteins, right? They also, if you see here, yeah, look at this, there's SSD proteins. The diagram is from your book, right? So these single strand binding proteins, they protect the single strand regions, okay, from degradation, because all this is happening in the nucleus. So we don't want these to be, degraded somewhere in the nucleus. So they kind of cover them and they're protected from degradation during replication. Excuse me. That was all. So knowing each and every enzyme is so very important, right? So you should know each and every, the function of each and every enzyme. Okay. And did you see the videos which I uploaded on Moodle? I uploaded almost like 8 to 9 videos. Did you all watch those videos? They're very interesting videos. Yes, boys and girls. Did you find the videos on Moodle? Hello? Are we connected, guys? Yes, you found them. Okay, wonderful. Yes, I'm going to upload a few more. Okay. So now let's try to quickly sum up everything. What are the basic rules of replication? Okay, the basic rules of replication are, which we already discussed previously. We know that replication takes place always in a semi-conservative way. One old strand, one new strand. It always starts at origin. Okay, so I've kind of made this slide so that we can sum up everything, in few sentences, and it doesn't go out of your mind. Okay, it gets, it stays in your mind. Okay, that's why I made this slide. So it starts at origin. Okay, we know that. And origin, we spoke about that. It's very rich in adenine and thymine, right? Rather than guanine and cytosine. Okay. And then I deliberately put a point here. It can be unique. It can be actually, see, we spoke about only bidirectional thing because that's what happens in the eukaryotes, but it can be unidirectional also, right? That is in the prokaryotes. Okay. So then it's semi-discontinuous. What is semi-discontinuous? Anybody? One strand is continuously synthesized, while as the other strand is synthesized in fragments, the Okazaki fragments, right? So that's called a semi-discontinuous, right? And then the synthesis will always and always take place in 5 to 3' direction, while as the strand will be read, right, in the 3 to 5 direction. Okay, and RNA primers are always required, right? So these were like the 6 very important points which you need to remember about the replication. Okay, any question till now?
Doctor, I have a question.
Quickly, yes, dear.
Are the clamp proteins the same as the topoisomerase?
Are the same as what?
I don't know how to say it, but the
which polymerase you mean?
topoisomerase?
No,
the clamp protein.
No, of course not. Of course not. Topoisomerase, I showed you the video. If you would have watched that video, okay, it removes the supercoils, right? This one is a topoisomerase. It removes the supercoils. While it's a clamp protein, something which holds the DNA strand, which clamps the DNA strand. Okay, that is what you see here. Look at this. This is a clamping protein. Okay, so it's holding the DNA strand. That's the clamp protein. While as you will find topoisomerase, you'll find it somewhere over here because there'll be supercoils formed over here. So it's going to cut and remove the supercoils. Okay. So the tangleness, because they're all tangled together. Okay, the positive supercoils. Clear, dear?
Yes, it's clear. Thank you.
So you'll find that somewhere over here. Okay, the topoisomerase. Because it works ahead of these, the replication fork. Okay. So that was about the, okay, now we'll talk about the PCR. Before I start PCR, tell me how many of you have heard this term before? And I'm sure that after what the COVID thing happened, and most of you are totally exposed to a term called RT-PCR test, right? Everybody says that, oh, the COVID test is being done by the RT-PCR. So do you know what is RT-PCR and what is PCR? Yeah. What is it about? Just tell me in one word, what happens in PCR? Polymerase chain reaction amplifies DNA. Okay. What else? Not really. Okay. Anybody else? Creates more copies of it for testing. Yes. See, now we are more kind of exposed to this term. I'm sure that before code you would have never heard this term before, unless and until you had studied molecular biology somewhere. Yes. So polymerase chain reaction is basically PCR. Is basically if you have a less amount of DNA, okay, and you want to amplify the DNA, okay. Say for example, let's say an RT-PCR thing, even though the RT-PCR is different, but the basic procedure remains same. Let's say when we give a sample for COVID testing, what do? They scrape our they take the sample from the mouth or from the nose. So the sample is so very less, okay? And for the COVID test, they need an RNA. It's an RNA-based virus, right? And RNA is automatically very, very small amount. DNA is more than the RNA in the cells. So imagine they need to work with the RNA, and extracting RNA in the lab is so very difficult. It's a really challenging process, right? To extract RNA in the lab, right? So imagine they have to isolate the RNA from that particular smaller sample which you give for the COVID, right? So what they do is that they use the PCR. They increase, they amplify the RNA, they amplify the amount of sample, right? So that they have enough sample, and they can actually find out whether this RNA belongs to the COVID. If this, because when a patient will give a sample, so the person who's testing, if the patient is positive, then the patient will have not only the mRNA from COVID virus, the patient will have his own mRNA also, his own tRNA also, right? But then he needs to amplify the amount of RNA, so that then he uses a probe to find out, okay, the markers to find out that, okay, whether this person is positive or not, okay? But in order to find out that, he needs to amplify the RNA of the COVID virus, right? Or even RNA of the, our RNA, from the particular patient, right? So in order to amplify that, to increase the amount of it, we use something called as a polymerase chain reaction, PCR, okay? Did you all understand what is the basic purpose? Okay, what exactly happens, in the PCR? Okay, you can think like, the way you have a copier machine, your Xerox machine. You have one paper, right? And you want to give it to 100 girls. When I prepare, for example, a paper for examination, I might have 200 girls in my classroom. What do? I prepare the master copy, and then what do? I put it in the copier machine. What happens? It starts copying it, like if I need 100 copies, 1,000 copies, 500 copies, right? That's what is happening. So that is the same thing, is basically your PCR. It's called a molecular photocopier, right? Because molecules are being photocopied. Their amount is being increased, okay? And it is an in vitro procedure. When I say in vitro, that means it doesn't happen in our body. We do it in the lab. Something called in vitro and in vivo. In vivo means in our body, and in vitro means in the lab, right? So we amplify the DNA to get millions and billions of copies of DNA, right? Within few hours. That is very important. Within few hours with very high precision, right? Very, very high precision, okay? Or sometimes, you know, uh, I go to for example, I'm a police woman, right? And I find that, oh, there's a murder which has been done in a particular house. As I reach to that house, I see a small blood sample, okay? And I suspect that this blood sample might be from the person who has murdered. What do? The blood sample is so small, and I don't have that person around me. What do? I simply take the dried blood sample, okay? I take it to my lab and extract the DNA from that blood sample, and then use PCR to amplify it. Do you understand what are the uses of the PCR, how important it is? Even if I have a single cell, my PCR can still work with that, right? Even if I have a very old tissue, like Egyptian mummies, okay, you can take the DNA from those Egyptian mummies and do PCR, and that's what people have done, right? And it is simply it simply increases the amount of the DNA. Did you all understand what is the basics behind the PCR? Everybody? It was developed by a very, very important person. I think everybody knows this person, like Carey Mullis. It was developed very recently, like 1983. He developed the PCR, right? And he got a Nobel Prize also for that. And what happened, the PCR is basically based on your DNA replication. It's simply the procedure of what we studied in the DNA replication. So the same thing, we do it in the test tube. That is the difference. That's how the PCR machine will look like, very portable, very small, very much with very good results, right? And it's simply whatever happens in the cell, okay, the DNA replication, we are copying the same thing in the test tube. Okay, what does that mean? That means if we just spoke about, we need a DNA sample, right? We need the parent strand of the DNA. So I have a parent strand. I have a sample which I took from the patient or which I took from that blood sample. So I need a sample, right? I need nucleotides, of course, right? In our body, we have naturally producing nucleotides, but in the lab, we can produce these, nucleotides artificially. So I use all the DNTPs, deoxynucleotides. I use a primer because we know that DNA polymerase will not work without the primer, but we don't have a DNA polymerase here because DNA polymerase is in the body. So what do we have here? We have here something called as a Taq polymerase over here. So Taq polymerase is exactly like a DNA polymerase, right? Which can extend the primer, right? But this Taq polymerase is so very important. In 1989, this Taq polymerase was declared as a molecule of the year. It is so very important because it can withstand the highest temperatures. In a while, I'll explain you, how do we do the PCR, right? So we increase the temperature up to 95 degrees Celsius, right? So we wanted a DNA polymerase which can survive at such a high temperature, and that is how we got this Taq polymerase. So PCR is possible just because of Taq polymerase, and as a result, it was declared as a molecule of the year, okay, in 1989. Okay, so how did we get to know about PCR? The Taq polymerase. We found this. It is also a DNA polymerase enzyme. Okay, very special type of DNA polymerase. Okay, which we got from a bacteria. Okay, so Taq polymerase we got it from a bacteria called as Thermophilus aquaticus. Okay, this bacteria lives in hot springs. You know what are hot springs? You know, guys, what are hot springs? Boiling water, right? So this, it stays in hot water, this bacteria, right? So as it stays in hot water, so it can survive in hot water. So that is why its DNA polymerase is also, can stay active in, at high temperatures. Okay, so that was the very important point. That was the key point, right? What was so special about this enzyme was that when I use my Taq polymerase in the PCR, okay, then what happens? It you know that after studying chapter enzymes, you know that how denaturation of enzymes, high temperature can cause enzymes to get denatured, like most of the enzymes, isn't it? But this is the only enzyme which can withstand high temperature in the PCR. And now after Taq polymerase, they have isolated many other enzymes as well, PFU, WENT, but this is like the best one. This works very good when you do it in the lab. So we understood what is a PCR, why do we do PCR, the purpose of PCR, and the requirements. What are the requirements of the PCR? We also saw the machine, how it looks like, right? Now we need to know how exactly do we do PCR in the lab. Okay. So this is a diagram from your book. Okay, so please pay attention. What do we do in the PCR? The same thing, it's like the same thing what we learned in in the DNA replication. Okay, so for example, the sample which I got from a particular patient, or the sample which I got from the blood stain, which was there at the murder site. Okay, what I did is I took that sample and with all these things, with primer, nucleotides, I put the sample in my test tube. Okay, I put the test tube into the PCR machine. As I put the temperature in the PCR machine, I set up the temperature here. Okay, so the temperature is basically like usually 95 degrees Celsius. Okay, it's a very high temperature. Okay, so what happens at 95 degrees Celsius? The two strands are separated. Okay, at such a high temperature, the hydrogen bonds between the two strands are broken and the strands separate. As you can see here, this is my old strand, this is my old strand, right? What do I do? Then the next the step 2 is basically, the temperature is decreased. Okay, you put a program in such a way that in the next step, the temperature decreases to like 70 degrees Celsius. There is a range basically for this, okay, but that varies, this we call it an anemic temperature. Anemic temperature can be between 60 to 40 to 60. It depends upon which sample you're working with, right? Let's say I use 60 to 70 degree temperature, right? So why use 70? Because that is what your book says, right? But I can use anywhere between 40 to 60. Okay, and here I can use anywhere between 90 to 98 temperature for denaturation. Okay, so in this step 2, what happens? Okay, I decrease the temperature, okay, to almost, let's say, 70 degrees Celsius. Okay, so what happens because of this decrease in temperature? We say that at this temperature primers are annealed. If you see here, look at the green ones. The primers are ready to start the DNA replication. They kind of hug the parent strand. Okay, so as primers are annealed, what is going to happen, right? So primers bind to their complementary sequences, right? And that happens at the temperature 70 degree, right? And step 3 is, like, that DNA extension. Then comes your Taq polymerase. It extends the DNA in 5 to 3 dash direction. It extends the DNA in 5 to 3 dash direction. We call it polymerization. So remember the steps, very, very important. Step 1 was denaturation. I use 95 temperature to denature my strands, to break the hydrogen bonds between the strands. Step 2 was annealing. What is annealing? That means my primer got, it hugged the DNA strand, or you can say, you know, it attached itself to the complementary DNA strand, right? And that happened at a temperature of 70 degrees Celsius. Okay. And the last step was the DNA extension or the DNA polymerization, which again takes place. You reduce the temperature further down at 55 almost, right? So this is the range which you can use for polymerization, but let's go with your book. So 55, let's say, okay. And then at this step, the nucleotides are added, and halas, I have at the end of my one cycle, okay, I started with how many DNAs, guys? I had one DNA molecule, right? At the end of my one cycle, I have 2 DNA molecules, isn't it? Then the same process will repeat. So many in cycle 2, you will again have denaturation, you will again have primer annealing, and you will again have DNA extension. So after my cycle 2, look at this. How many? I started with 2 in cycle 2, right? I have 1, 2, 3, 4, right? At the end of cycle 2, I have 4 molecules of DNA. Remember, I had only 1 molecule. At the end of cycle 2, I have 4 molecules of DNA. So cycle 3 will keep on repeating. The same process will keep on repeating, the denaturation, annealing, and extension. At the end of cycle 3, I have how many? 1, 2, 3, 4, 5, 6, 7, 8. 8 molecules of DNA. I started with just 4 molecules of DNA, right? So these cycles keep on repeating. The way you put your clothes in the washing machine, and cycles keep on repeating, right? Same thing, these cycles keep on repeating, and at the end of maybe 100 cycle or 50 cycle, right, you can get huge amount of DNA depending upon your need, what is your requirement in the lab. This is called as a DNA polymerase. This is called as a PCR. Yes, temperature is so very important, so very important, because we rise the temperature, then we reduce the temperature, then we again rise it a little bit further, right? So it's all about, you play around with the temperature, right? And that's how you get the, uh, you know, at the end you get the huge amount of DNA, right? So I just, wrote the steps here so that you don't forget it ever, right? So denaturation, primer annealing, and extend. It was not there in your book, but yes, we do ask many questions based on PCR. So you have to know the temperatures, you have to know the steps, okay? What all happens. Are we okay? Did we all understand? So did you all watch the video which I had uploaded on Moodle for PCR? You want me to show that video to you now? Or you'll watch it yourself on Moodle? Okay, you want me to show that? Okay, great. I hope it will work. I'm just kind of, yes, Hannah, I will, don't worry. Okay, give me a minute. I hope it will work, hopefully. Yes. Tell me if you're not able to hear the sound, or if you're not able to hear the sound and video. Just switch on your mics because I will not be able to see your chat window. So if you're not able to hear the sound or see the animation, just open your mics and let me know about it.
Doctor, there's no sound, only the video.
Only the video?
Yeah.
Okay. Give me a minute. What happened? Wait a minute. I'm just trying to play it once again. Shared video. Screen sharing has failed to start. The window you selected is invalid. Why? It's not an invalid window. Show all windows. I am. Okay, nice. 1 minute. Okay, I think we should be able to hear it now and watch the video at the same time.
Reaction, or PCR, uses repeated polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Yes, ladies, how was it? Did you all understand now? Is it clear now? Trying to upload the presentation once again. As I said that it sometimes behaves, you know. Can you see the screen now, ladies? Is the screen visible? Okay, good. Alhamdulillah. All well. Okay. So is it clear? Yeah. So it is like, you know, it has changed our life. I'm going to upload a video, you know, how they're doing the COVID test. The COVID test is based on the same, exactly the same, you know, procedure, but we use a different PCR because now we have plenty of PCRs available, different variants available. Okay, so we use an RT-PCR for that. Okay, because here in the COVID virus we have mRNA. Okay, as a genetic material. They don't have DNA as a genetic material, so we need to convert RNA first to the complementary DNA and then do the PCR. So the RT-PCR is specialized for that. I'm going to upload that, on Moodle, and you can just watch that. Okay, that's going to help. That's going to be very helpful, right? And nowadays, it has totally changed the whole scenario. Trust me. Once the COVID started, within just few days itself, they standardized the procedure how to detect the COVID virus in patients. Similarly, it has been used everywhere in medicine, whether we talk about DNA fingerprinting to find out who is the parent, for a particular child, if there's an issue that, basically between two parents, that this child belongs to me, and this, any dispute between two parents. So they do DNA fingerprinting to find out that, or to detect any tafra, any mutation, cancer research, sequencing the DNA, the Human Genome Project. We'll talk about that in next chapter, right? So all these things were possible, or diagnosing so many genetic diseases. Oh my God, it is so helpful for all that, right? And as I said, that COVID test is also based on the same technique. Okay, moving on further quickly. Who will tell me the answer for this question? Nucleotides are added to the growing DNA strand during which step? D, extension. Thank you, Mohammed. Absolutely. Thank you. Extension polymerization, right? Step 3. So during annealing, what happens during annealing? And remember the temperatures as well, everyone. Okay, stick to the temperatures present in your textbook. Annealing, what happens in annealing? Yes, Hannah, absolutely. The primers bind to the newly separated strand. This is the answer. Absolutely, very good, right? So this is the last topic for this chapter, okay, which is basically how DNA is repaired. Okay, now that we know that DNA polymerase has synthesized the DNA, we have the huge amount of DNA that, but is that DNA okay to be sent to the next daughter cell? What does that mean? That means, do we have any mutation in that DNA? That is very, very important. Before that DNA can be incorporated in the daughter cell, it's very, very important to find out if that DNA is all okay, if that DNA is mutation free, right? So DNA polymerases not only build the DNA, but they also are experienced in checking their work, right? For example, if you write an answer before submitting the paper, what do? You check for the spelling mistakes, right? Right. So we call it proofreading, right? So DNA polymerase is such a marvelous enzyme that it not only builds the DNA, but it also checks its work before it can actually finalize that, okay, the DNA strands have been formed, and that's called as a proofreading. And what happens in proofreading? Any wrong base. Suppose I had here in my DNA, I had here adenine, thymine, and guanine, let's say. And let's say a new strand of DNA which was synthesized, okay, let's say this was a new DNA strand synthesized, and I had here what? What should be here? Adenine base pairs with thymine, right? Thymine with adenine, but unfortunately I don't have adenine, I have a guanine here, right? And let's say this is cytosine, right? That's how they form the bases. But adenine and thymine, all good. Guanine and cytosine, all good. But I have a mutation, I have a tafra. And where is it? And it's basically at this particular point. So DNA polymerase made a mistake. Instead of incorporating what? Adenine over here, it made a mistake that it incorporated guanine over here, right? So that wrong base, right, which was added accidentally by DNA polymerase, that will be removed and replaced, okay, before continuing further. Very, very important, right? So even though the errors made by the DNA polymerase are very, very less, right? But yes, sometimes there are errors in the DNA which is made But at the same time, as long as your DNA is able to, your DNA polymerase is able to proofread it, right? Then, and your DNA, there are many DNA repair enzymes in our body. As long as, alhamdulillah, we have no problem with the DNA repair enzymes, so homeostasis will be achieved, right? So any mismatch repair can be removed and the new base will be incorporated. But God forbid, if there's any problem with our repair mechanism, then the mutation will be there, right? There are plenty of DNA repair enzymes, but we'll only stick to those which are present in your textbook. The two main important repair methods are your base excision repair and the nucleotide excision repair, right? So excision means what? Removing, knocking out something, right? So if you see here in the BER, okay, what happens? The base is being removed. Only one base which was incorporated in a wrong way, that is removed. While as in the NER, the nucleotide which was, incorporated, in a wrong way, the whole nucleotide will be, the whole chunk will be removed from the DNA. Let's try to discuss the BER first. Okay, so please pay attention. So you remember this, we spoke about, beta globulin gene, right? What happens here? Instead of having what? Thymine over here, the triplet should be CTC. What do we have? We have adenine over here, right? Guys, do you remember this? Sickle cell anemia? And then we have valine. And look at it, just one base pair was changed and it changed everything in a particular person, right? So this should be removed by the base excision repair, BER, for example. Now let's try to learn the process, how it happens basically. So step one, what happens if the if DNA polymerase had made a mistake? Okay, what it's going to do? It's going to it has an enzyme called as a DNA glycosylase. Remember all the enzymes. Okay, it has an enzyme called as a DNA glycosylase. What is the function of DNA glycosylase? It scans the whole strand, like a scanner. Okay, for example, this is our DNA strand, and the mistake done by the DNA polymerase was that it has put uracil here. Do we have uracil in DNA replication in the DNA strand, guys?
No.
Absolutely not. We don't need uracil, right? Uracil should be in the mRNA. It should not be there in the DNA. So it was totally a wrong mistake done by the DNA polymerase, right? So G glycosylase will simply scan, like, oh, mistake found. Okay, it looks for this mistake, looks for this damage. Okay, it recognizes it and then removes this damaged base. How? If you remember, we spoke about the glycosidic bond is present between the base and the sugar. Remember, it simply breaks this glycosidic bond. Khalas. Okay, so the glycosidic bond between uracil and the deoxyribose sugar was broken, right? So as it was broken, as you can see here, the uracil is leaving DNA now. Okay, so don't you think so there will be a gap created over here? Isn't it? So that gap, if you see here, look at this, there's a gap created over here, right? So that gap which is created is called an AP site. What does AP means? Remember all these points, very, very important. What is AP site? AP means epurinic or epyrimidine site. That means there's neither a purine over here, there's not a pyrimidine over here. So this site will be later on called as the AP site, AP. There's no purine, there's no pyrimidine. Apurimic, apyrimidic site, right? So what happens, the next one will come, the next thing is there's one more enzyme which will come, which is called as an endonuclease. Remember the enzymes, right? So what will endonuclease do? Endonuclease will simply degrade the backbone now, because we want to remove this backbone as well, okay? So it will remove the phosphate as well as the sugar. That will be done by the exonuclease, okay? But then there's a difference between exonuclease, exo and endonuclease, okay? If this nucleotide is present at the end, at the periphery, so we have an exonuclease. And if the nucleotide was in the middle, then we have an endonuclease. That is why they have used the word endo here, okay? But if the same thing would have been over here, Then the same enzyme, but we call it exo, which can cleave or which can remove the nucleotide, the incorrect, sugar phosphate base pair, okay, at the end. So that is the difference between endonuclease and an exonuclease, okay. And now what will happen? Which enzyme should come now? We remove the base, we remove the sugar phosphate backbone. Which enzyme should come now, guys? What do you think, girls?
DNA polymerase?
Absolutely. We should have a DNA polymerase, and DNA polymerase is going to incorporate a fresh new nucleotide, okay, which was supposed to be over here, right? And then the last enzyme which will come is the what? DNA ligase, absolutely, which is going to seal the gap, like stick all of them together, stick it together, right? That's how the whole thing will be repaired. Hannah says, so can we say that if someone has any mutation in the DNA, it means that they lack? Yes, there are many reasons. One whole lecture, can be given on DNA repair mechanisms, but that could be one of the reasons. That means if somebody's suffering with a cancer, right? That means, like, there are many, 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 many reasons, but this could be one of the reasons that their repair enzymes have not worked the way they were supposed to. Yes, Hannah, you're absolutely right. Right? And all I want to tell you that is, this is a diagram which is present in your textbook. And trust me, all my students, they get lost when they see it. They don't understand anything in this diagram. So no need to prepare this diagram for the exam, for the midterm 3. Prepare this diagram. So this can help. This is like a more easier version of this. This looks kind of very scary. What do you think? Yes or no? Yes, so for the exams, you can prepare this one. This is pretty much easier than the other one, okay? So even though it's from the textbook, I know it looks with so many structures. It's the same thing, okay? But here, it's just that we have structures over here, and students, they tend to feel that structures are very difficult. So that's why this one is like the minimum structures I could have here, okay? And the last thing is then the nucleotide excision repair. As I told you that here, the whole nucleotide is removed rather than just one base pair, right? Sometimes, DNA polymerase makes a mistake, not even one base pair. Almost 20 to 30 nucleotides have gone wrong, okay? So at that time, the same thing, right? The procedure is same, right? It's going to remove the damaged nucleotides and replace those nucleotides with the new one, okay? And this usually happens, this usually works when we go in the sun, you might have seen that, you might have heard about if you if you take a sun bath for a very long time, if you stay out in sun for a very long time, okay, it might damage our DNA, right? It might cause cancer, right? The reason for that is whenever we go out in the sun, because of the UV rays, okay, there are a lot of thymine dimers which are formed in our body. Now, usually, that adenine base pairs with thymine, right? And guanine with cytosine. So when we go out in sun, right? So thymine, the two thymines which are present nearby, this thymine and this thymine, they form a bond between, with each other, right? So when we stay out in sun for a very, very, very long time, right? So it results in the formation of thymine dimers, and those, when we get a sunburn, right? A suntan, okay, when we're exposed to UV radiations, okay? So as long as, alhamdulillah, we have this nucleotide excision repair, right? We're all okay, because these enzymes, they're going to remove all these thymine dimers, okay? So, or even when, when we're exposed to the X-ray radiations, you might have seen that when we go for the X-ray radiations, it's better to avoid the radiations. And even the person who's doing X-ray, he stays away, okay? He stays in that corner room so that, he's not exposed to the radiations, because it can also form the thymine dimers, okay? Now, one of the examples which they've given is the xeroderma pigmentosa, okay? This is basically an inherited disease where the patient is lacking the NER repair enzymes. He does not have NER repair enzymes. and it is an autosomal, genetic recessive genetic effect, okay, wherein the DNA is mutated, okay, and as a result, the person becomes more and more sensitive to UV light, okay. So as at the moment they go out in the sun, right, so they develop these pigments, as you can see his face, right, they develop these pigments as spots, tumors on the skin, okay. So this is like a severe form of a sunburn which they get, okay, because they do not have NER repair pathway. Now you should be so thankful to God that we are all, alhamdulillah, right, with everything intact, right, with everything blessed. So we should every time be thankful to God, isn't it? Look at, he just doesn't have one enzyme. Imagine, the entire life goes for the toss for this particular person, right? With that, yes, definitely thank our God, definitely, no doubt about it. With that, we finished the chapter, and I just wanted you to recall so that you don't forget the name of the enzymes. Name all the enzymes in the base excision repair. So it's the DNA glycosylase, it's the AP endonuclease, it can be an exonuclease as well, then the polymerase to extend the DNA, and then finally to seal the gap with the DNA ligase. And the last thing which I put here so that you don't forget here, what is the sequence in which these protein functions or enzymes function during DNA replication? I should be using the word enzyme here. Enzymes. Who will answer this? Who will answer this? Tanya, yes dear, please go ahead. Please number them as per the order.
Is helicase the first one?
Very good. It can be helicase, it can be even topoisomerase, but I can take that as well. It can be even this number one. So then number two?
RNA primase?
I said that. It can be either helicase number one or topoisomerase number one. So let's put topoisomerase number two. You can stop now. I will ask somebody else to answer now. Thank you, dear. Mohammed Suleiman Javed. Yes, Mr. Javed. Can I have the answer for this question? Okay, thank you, Mohammed. Thank you, Abdulaziz. But the question goes to Mr. Javed. Primase. Thank you, Suleiman, for replying. Thank you. Okay. Yes, wonderful. So this will be number 3. Very good. Okay, let me... Shaima. Yes, dear. Shaima, which one will be number 4? Ligase or polymerase? Thank you, Mohammed, for replying. I need Shaima to reply. The shield says polymerase. Shaima, primary? What is primary? It's the polymerase, right? Yes. Where is my pen? I lost my pen. Okay, here it is. So this would be my number 4, and this would be number 5. So this could be like, even this could be number 1 and this could be number 2. That's perfectly okay. Even if it's a vice versa, it's perfectly okay because they all work together, right? So yes, this is it. Okay. With that, we finish the chapter. Alhamdulillah. So we still have half an hour left. We'll take just a short 5 minutes break and then we can start with the next chapter. As I said that we're kind of lagging behind. We were supposed to finish this chapter last week. We're almost 1 week, we're delayed, Okay, so just take a quick 5-minute break and then we'll come back for next chapter. Meanwhile, I will upload the chapter. Okay, just take a quick coffee, tea, water break. Thank you. Okay. So it's pretty much related to the previous chapter, because in previous chapter we learned that the central dogma is, your DNA forming mRNA, okay, and mRNA forming the proteins, okay. So before DNA can form mRNA, DNA also needs to replicate itself. So previous chapter was based upon the structure of the DNA RNA and then how replication would take place. And this chapter is entirely based upon how transcription and translation takes place. Okay, so even though things are not in a very detailed manner over here, they've combined the transcription and translation, but whatever is there in the book, okay, we'll just try to learn all that. Okay, we'll not go into very much detailed way, right? But yes, we'll try to learn what is the transcription and the translation, like more like a superficial one. Okay. So as I said, that the central dogma is DNA forming mRNA, irrespective of any RNA. We're not saying only mRNA, right? Because only mRNA forms the protein. But by the end of the day, all the RNAs are made by the DNA. So this is like a main confusion which students always have. They think that DNA only forms mRNA. That's not the case, guys and girls. It's the DNA which forms all the types of mRNAs, whether we talk about tRNA or we talk about ribosomal RNA. But when it comes to protein formation, we say that it is the mRNA which will be further converted into or translated into a protein. Okay. So, and that's how a gene is expressed. That's how a protein is expressed in our body. Okay. So before we start, we first of all we'll try to know what is the difference between replication and the transcription, right? We recently spoke about DNA replication. It's very fresh in your mind, right? We saw that in DNA replication that a primer is required. You'll see here the enzyme which we use here is the RNA polymerase, not the DNA polymerase. So RNA polymerase does not need primer at all. Okay, this is like a first basic difference here. And in the DNA replication, we had adenine forming a bond with the thymine, okay, and the guanine with the cytosine. Since we know that there's no thymine in the mRNA, so the thymine will be replaced with the uridine or uracil, you can say that. Uracil is the base, right? And then we know that in the DNA replication, very important, we used both the strands to replicate, right? We used both 5' 3' end as well as the 3' 5' end to form the replication. The entire genome was replicated. But you will see here, no, we use only one strand of DNA, the template strand, also called as an antisense strand, also called as a minus strand, which will be transcribed, which will be converted into the mRNA. Okay. And then the next one is only a portion of the genome is transcribed. That means the entire DNA is not converted into the messenger RNA or the RNA. But we saw in the replication that the whole genome is copied, right? That's such a big difference between the two. Okay. And of course, there we had the deoxynucleotides and here we have the ribonucleotides, right? The sugars differ in the DNA and the RNA. Okay. And one of the most important differences is like at the end of the replication we get a double-stranded DNA, right? But you will see that at the end of the transcription we will just get what? One single-stranded RNA. A very important difference. Yes, Mohammed, you're absolutely right. Transcription is same as replication, reading from 3 to 5 and writing from 5 to 3. Yeah. So transcription also takes place from 5 to 3' end. So that is like a similarity between the two. Okay. Thank you, Mohammed, for raising this question over here. Okay. Then I made a table over here so that you can differentiate it further. DNA polymerase, RNA polymerase. Okay, on chromosome in S phase, like selected fragment of DNA. RNA primer is required, no primer is required. Both strands are used for the template, only one strand is used. Proofreading here is done by the DNA repair enzymes and by the DNA polymerase, right? The proofreading by the DNA polymerase. Okay, here you'll see that there's no proofreading for the RNA polymerase. Okay, and then here we have the deoxyribonucleotides, and here we have just the ribonucleotides, right? This is like a basic thing which we should know. What is the difference between transcription and the translation? Okay. Now, one of the most important questions is that, where does transcription take place in the cell? Remember that transcription always takes place in the nucleus, because DNA is there in the nucleus. The big boss is in the nucleus, right? So the DNA does not come out of the nucleus. So obviously, transcription has to take place only in the nucleus, which is very, very important. Okay, and we already spoke about transcription as the process by which the information encoded in the DNA molecule is copied, and now the mRNA has a recipe of making protein. The recipe of making protein was actually with the DNA. Then what DNA is doing, DNA is giving that recipe to mRNA, and then mRNA is converting that into the protein, right? So that is the transcription. Okay. And then how many RNA polymerases do we have? Okay, we have three types of RNA polymerases. Okay, we have RNA pol 1, we have RNA pol 2, and we have RNA pol 3. Pol 1 forms the ribosomal RNA, pol 2 forms the mRNA. So we'll be mostly working with this, okay, because we're going to talk about only mRNA formation in this chapter, right? So pol 2 will form the mRNA, and pol 3 will form the tRNA. These are the three basic RNAs which we need for the protein synthesis. Okay, and then you will see that there's something called the transcription factors. Okay, which is, I think I forgot to mention that difference here. I think we should put one more difference here, that in replication, DNA polymerase does not need anybody's help, does not need transcription factors, okay, to start the replication. But you will see here RNA polymerase are, they're like blind. They cannot see, and they're not blind. I'm just kind of, trying to tell you that, that's how you can remember it. RNA polymerase need help of something called the transcription factors, which we'll talk about in a while, right? So they definitely, these transcription factors, which control actually the conversion of DNA and the mRNA, right? Very, very important, right? So transcription regulation is done by the transcription factors, but there's no question of transcription factors in the replication. You all can add this point in this slide as well as in this slide, okay? No transcription factors required in replication, while as yes, we need transcription factors in the, what do you call it? Transcription. Okay, and we're going to talk about what are transcription factors in a while. Okay, are we okay till now? Any questions till now? Okay, no questions. Okay, now one of the most confusing things which I've seen always, students do get confused, so please pay attention. Okay, so as I said that in transcription, both the strands are not used for the transcription. Out of two strands, only one strand will be used for transcription. Okay. Now the strand, now please, everyone, please pay attention. The strand which is used for the transcription, which acts as a template for the transcription, okay, that strand is called as a template strand. So in this diagram, as you can see here, this is a template strand. Now this template strand has many names, and you need to know all the names. It is also called as a minus strand, just the word minus, okay. It is also called as an antisense strand, okay. And now what about the other strand? So template strand is that strand which actually forms the mRNA, okay, while as the other strand which does not form the mRNA, that is called as a coding strand. So whenever we give this question, student, if I give the option of template as well as coding, student think that coding means, okay, that means this strand is forming the mRNA. So remember that it's not the coding strand which forms mRNA, it's the template strand. Now, why do we call it a coding strand? Because the new mRNA which is formed over here, this one It has the same sequence as this one, except here we will have uracil, right? And here we will have thymine. So that is why the word coding strand has been given to this one, because this and this will be have same bases. Why this is called an antisense, because the new mRNA and the old DNA strand, the template strand, their base pairs will be different. If I have thymine over here, right? What will I have over here, guys? What will I have in the mRNA if I have thymine in the template strand? I will have adenine over here. No, we'll have adenine. Like, absolutely right. We'll have adenine over here, right? So that is why here the base pairs are complementary, right? So that is why this is called as, this one is called an antisense strand, but this is a template strand just because it forms the mRNA. Okay. So what are the other names of the coding strand? The other names are, this strand is also called as non-template strand. Non-template strand. This strand is also called as a positive strand. Let me write it over here. Positive strand. Okay. And that is it. Okay. And yeah, and sometimes it's also called as a sense strand. Yes, you're right. I mean, I'm right. It's also called as a sense strand. This is an antisense. This is a sense strand. So these are the different names which we use for the other strand which does not form the mRNA. I repeat, coding strand is the one which does not form the mRNA. We also call it a non-template strand or a sense strand. Are we all okay with this? Did you all get it right? Yes, Dana, I just said that. Yes, I just said that the strand which forms the mRNA, that strand is called as a template strand, that strand is called as a minor strand, that strand is called an antisense strand. Just remember the names. That's very important. That's all. Okay. And the other strand which does not form the mRNA, that's called a coding strand or a non-template strand or simply the sense or a positive strand. Just remember the names. That's very important. Okay. Okay, then we move on further. Okay. Now the question says that which strand encodes the RNA? Is it the template or the coding strand? Who will answer this question? Which strand encodes the RNA? Is it a template or a coding strand? Coding, huh?
Template.
Very good. It's the template, not the coding. Remember that. Okay. So it's the template one which forms the RNA. Okay. We move on further, right? As I told you that in RNA, the entire DNA is not used for the synthesis of the mRNA. We use only a small part of DNA. Let's say the DNA is from all the way from here to here, but we do not use the entire DNA. We simply use maybe a smaller part. Maybe this part is only used for the synthesis of the mRNA, right? Okay. So now I want everyone to pay attention over here. Now, if you see here, this is basically this is the the whole strand has been given over here, right? This is like your template strand. Okay. Now, this whole template strand, for example, is divided into two parts. Everyone, please pay attention. One is called as a structural gene. Another one is called as a regulatory gene. What is the difference between the two? Okay. Structural gene is only maybe from this to this. What do you mean by structural gene? Structural gene is that part of DNA which actually forms the RNA. So that means if I see this whole strand, which is basically the diagram which is on your slides, the whole strand does not form mRNA. It is only this particular part which forms the mRNA or any RNA. So this is called as a structural gene. Very important. right? So this gene might have exons, it might also have introns, as you can see here, because eukaryotic genes are not continuous, they're discontinuous, right? So I have an intron here, I have an exon here, and intron here, exon here, right? So this is called as a structural gene. Okay. So this structural gene, you will see that it might have an initiation site here from where the transcription will start. Okay, we'll discuss that once again later on. It might have a part here which is not overshown here. It might have a part where the transcription stops. Okay, we'll discuss that later on, the start and the stop signals. Okay, so that is about the structural gene. Okay, so what is a structural gene? Structural gene is that part of the DNA which actually forms the mRNA or which actually forms the RNA. But then this gene does not work all by itself. It needs the helper one. It needs the helper gene as well, or a helper DNA as well. So what is a helper DNA? The helper DNA is a regulatory gene. What is a regulatory gene? Regulatory gene is basically, for example, let's say this part is a regulatory gene. Let's say this part. Okay. So regulatory gene is that gene which controls this structural gene, or you can say which controls the transcription. So the question says that which of the following controls the transcription? Is it the structural or the regulatory? The answer should be it is the regulatory one. Is this regulatory gene transcribed? Can this gene be converted into RNA, guys and girls? The regulatory one. Can it be converted?
No.
No, never, 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 ever. Its job is not to be converted into mRNA. Its job is simply to control this part of DNA, this part of gene, which is actually being converted to the RNA. Remember this. So it controls the transcription. Okay, it's not transcribed at all. But then this part, like this regulatory gene, has a very important sequence here, as you can see here. Let me delete all this now. This sequence, which is called the Tata box. What is a Tata box? Okay. So the sequence on my regulatory gene, for example, in eukaryotes, okay, it has thymine, adenine, thymine, adenine, adenine, and adenine. In short, we simply call it a Tata box. Okay, we call it a Tata box basically in prokaryotes, but in eukaryotes we call it a Hogness-Goldberg box. But you don't need to know that. Just remember the Tata box as per your book. Okay, so what is this Tata box? Okay, Tata box is basically the one which acts as a promoter. Have you ever heard this term before? What is a promoter, guys? Have you ever heard this term before? What is a promoter? Anybody? Nobody? Okay. Okay, no issues. So what is a promoter? Promoter is basically that part, okay, promoter is that part of your regulatory gene where the, where the transcription, which are recognized by the transcription factors, okay, or you can say where the DNA polymerase, RNA polymerase binds to it. That is basically called as a, no, it doesn't stimulate. Promoter is basically a region, okay, on your regulatory gene, okay, which is recognized by certain proteins called as the transcription factors, okay. And once you have transcription factors binding over here like this, we'll talk about that again, what are transcription factors, okay. What happens after this? Then the RNA polymerase, when it sees that, okay, transcription factors are there, that means I need to start the transcription at this point. So this marks, like, this is a binding place for the RNA polymerase. But all these things did not happen in the replication. We never had transcription factors in the replication. So transcription factors are the special proteins which bind to the promoter. When they bind to the promoter, they further attract the RNA polymerase. So RNA polymerase can also bind over here. And then the transcription will be started. Are we okay? I think we'll stop over here because I have a next class. Okay. So did you all understand the structural gene, regulatory gene? This is so very important. And transcription has only this thing, you know. You need to know about only these things in the transcription, right? Yes, Rand, please go ahead. You have a question? So we'll continue with this in our next class on Monday, okay, next week. Yes, Rand, I've already posted the presentation there on Moodle. Or you want this updated one, Rand? Yes.
Yes, updated, please.
Yes, dear. I will do that. I will do that. Thank you so much. It's interesting, is it? Very good. Alhamdulillah. So we'll continue with this and we can revise what is a structural gene, what is a regulatory gene, and then we'll talk about in next class what are transcriptional factors, right? And that's the main thing we're going to discuss in the transcription here.
Thank you, Doctor. Have a nice day.
Thank you, you all. Thank you, everyone. Bye-bye.
Thank you, Doctor. Have a nice day.
You too. Bye-bye, everyone. I'm going to log off because I have a next class. Dana, you know that you have to send an email with, please send an email with the course you're talking about. Is it PHSF or PBio, with the section as well? Okay. Thank you, dear. No problem. Send me an email for that.